Welcome to Biology Lab Part 2, I suppose. Here with my esteemed colleague, Ms. Karen Rose, and we're going to shoot a quick video on biology.
And this is Mr. John Hopper for my students who haven't met him yet.
Oh yes, indeed. We're gonna have a great time today, and we're practicing our social distancing as best we can given the circumstances, and we're gonna have a good time, right?
Absolutely.
All right. So here we are. We're going to do Macromolecules Laboratory number 2. So I've got writing all over this thing, you know, just to kind of keep me where I want to be. You should have your lab book out and be making notes as well. It'll make your life much, much easier. Okay, so what we're going to be doing here is running some chemical experimentation, so to speak, so that we can identify specific macromolecules in solution. So imagine a world where you're presented with some sort of solution. You need to know what's inside of that. What we're going to be looking for are things like carbohydrates and lipids. In other worlds, we could do nucleic acids, you name it. There are ways in which to identify these things, and we're going to be showing you some of those today. Now,
so when you do this, today you're going to watch us make a lot of things, and we'll spend time in preparation, but we need to focus on the results at the same time, because when you go to have your lab test, the colors are going to be what's important to you. You won't be making things up. They will be made and you'll be shown test tubes. So pay particular attention to the results, to the colors that you get.
Now, to do this, imagine you're presented with this solution and you have to identify what's inside of it. You can't just have the solution and run the test and hope for the best. What we try to do here with scientific experimentation is have methods of adding control to the situation so that we know, given the terminology, for certain to some degree, what we're doing is correct and we have identified things in a correct fashion. So what we're going to do is, for all of our experimentation today, we're going to display what's referred to as positive and negative controls. All right? Positive and negative controls, which are very important for scientific experimentation.
And it works that when you have certain food samples like we're going to use today and they have colors to the food, then you're not confused because the food color is, let's say, purple onions would give off a more purple color anyway, then you know how they fit into that scheme. So the question is not so much, is it exactly the color of the positive control, but does the sample, is it closer to the positive or closer to the negative?
So let's imagine we are looking for proteins. We have a solution and we're not sure if it has proteins in it or not, but we're going to find out by running one of these experiments. What we will do is we'll have a negative control, something like water, for instance, that we know has no protein in it.
Absolutely.
Or if it does have protein in it, we have major problems with our water supply. And then we will run a second experiment with something that we know for absolute certain has protein in it. All right? What we're doing there is we're having a negative control.
And a positive control.
With our negative control being water, which we know has no proteins in it. Our positive control being something that we know for certain has proteins in it. So now, I've got negative control, which is water, in this case, with no proteins. I've got a positive control, which we know for certain does have proteins, and then we have an unknown solution.
That's right. So those other solutions, when you answer your questions, they're not controls, they are test samples. Because going into the experiment, you do not know if it contains a protein or not, or if they contain a protein or not. So they are test samples. You only have one positive control, one negative control per experiment.
All right, so that would be positive and negative controls. The first of our macromolecules that we're going to be dealing with will be the simple sugars, and for that matter, let's just say carbohydrates. So simple sugars and complex carbohydrates. What we'll be looking for here is simple monosaccharides like glucose and galactose and fructose. These could all be easily identified in solution given the appropriate chemical reactions, and we'll also be dealing with some polysaccharides.
So that means that we're doing two tests because the simple sugars are tested with a test called Benedict's, and here's the reagent. You can see the color of the reagent. But the complex polysaccharides, such as starch, are dealt with a different test with iodine. And I don't know if y'all can see, but iodine has a yellow-brown color. Okay, so we have two tests for carbohydrates.
So what you got to keep in mind here is these simple sugars are very small, just single ring structures for the most part, whereas some of these polysaccharides can be, hundreds of monosaccharides all bound together.
Hundreds of rings.
Very, very different. Very, very different. Now, What we will be looking at specifically for polysaccharides today will be starch. This is manufactured by plants, potatoes being the classic example. Whereas we, as humans, we don't make starch. What we make is glycogen in the liver, for instance. So we are capable of making and utilizing these polysaccharides as well. This is not just a plant thing. Just in our case, starch is. Okay. So the way in which we do this, as we established previously, is with this Benedict's reagent and an iodine solution as well. And those will come up with a color change when placed in if these macromolecules are present. And we'll be showing you that here in just a little while. The next portion of this experiment today is going to be dealing with lipids. Now, lipids are incredibly important to the human body. In fact, most of our cell membranes are lipid composed, okay? Like phospholipids, which we have heard of in lecture probably at this stage. Now, lipids themselves are hydrophobic, meaning
they are nonpolars. Remember when we talked about types of covalent bonds and electrons being shared equally? Those were nonpolar bonds, nonpolar molecules. And we use that word hydrophobic with that. Remember, they don't like water, so they try to get away from water.
It's like Grandma used to always say, right? Oil and water, they don't mix. And the reason oil and water don't mix, Grandma, is because of polar versus nonpolar molecules. Oils being considered a nonpolar molecule, whereas anything that is dissolved in water would be considered polar. Or water molecules themselves together would be considered polar. Sugars
are also polar, tend to be polar.
So what we're going to be looking at today is variations between these nonpolar hydrophobic
interactions.
Sure. Okay, I can live with that. And polar hydrophilic compounds, let's see. Now, also in your book, it just has it in there, so I have to talk about it, is the concept of an emulsifying agent. Let me give you an example. Okay, let's say I go out and I work on my car. I get oil all over my hands, and then I go into the sink and I go and I wash my hands with water. What's going to happen?
It all beads up. Remember we talked about hamburger grease in a pan?
It's just going to sit
Same
there,
thing.
okay? But what I can do is I can get something like my Dawn dishwashing detergent or something along those lines, and I can wash my hands, and that oil just sheets right off.
Because soap actually is able to mix with the nonpolar end of the molecule and then the polar end. Okay, so like soap has both polar and nonpolar, so it can mix with either one. Mixes with the water.
Two opposing ends of the molecule.
That's right. It will mix with water and the oil or grease, whatever it is on your hands.
So this is considered an emulsifying agent, which is not unlike the bile released by the liver, okay? It's very difficult for the body to absorb lipids if those lipids are not dissolved in an aqueous solution. And bile does that for our system so that we can then import those lipids into our bloodstream, which is very important for us. Now, today...
Okay, so we have Sudan 4. I think y'all can see that. Notice this is a red solution, okay? So a little different color than you saw with the Benedict's reagent and the iodine for the starch test. Okay.
So a very important concept to recognize when we're doing this Benedict's is the way in which the test tubes look once the test has been run. So if you're running this, here's two test tubes. I assure you that's what this is. And here is our lines at the top. This is air and this is liquid. When you run a Sudan test on this, what you tend to end up with is this is going to be a solid, that's a dyslexia, solid color if this is a negative. And if it's positive, what you're going to see is there's going to be like a line like this. This is going to be kind of a pink, and this is going to be more of a deeper red.
So the negative will be a solid red, as Mr. Hopper said. Just water will be red, okay? But you'll see a separation like you do with salad dressing with oil and vinegar, something like that. So notice that the colors that Mr. Hopper drew tend to show up as more time, as the test tube has settled and more time has gone by. So when you initially make your test tube, you'll see some layering. And then if it sits there 5-10 minutes, something like that, you'll start seeing the color differentiation. And actually, if the test tube's set for like an hour or so, all of the Sudan 4 would move out of the water layer into the oil layer. And you would see this is totally clear, like water should be.
Lipid positive, lipid negative.
Exactly.
All right, perfect. All right, so the third part of this little experimentation today is going to be on proteins. Now, what you need to consider here before we go any further is that you are quite familiar with the concept of a protein and what a protein is. When you see someone, okay, your hair, your nails, your skin, your musculature, I mean, it's protein.
Exactly.
Okay, it's protein. So having experimentation that gives us the capacity to determine the presence of these proteins is incredibly important. And before I go any further, I want to get a term across to you, and that is the concept of a peptide or a polypeptide or a peptide bond. Now, the building blocks of proteins are amino acids.
Sure.
And what we need to consider is that these are going to be, in essence, covalently bonded together. But we don't say covalently bonded per se. When we're dealing with proteins, we say peptides.
Oh.
Okay, okay, we say peptide. So anytime you hear the term polypeptide or peptide bond, you need to be thinking protein. Okay, you need to be thinking protein. Yeah, I'm pretty happy.
So sometimes, as they refer to in your lab book, they make reference to, would you have a peptide? Would you test a peptide? And while we won't be doing that today, peptides are shorter and they don't have their own function. So when, like Mr. Hopper said, you see somebody and you think of their characteristics which are made of proteins, you're thinking protein. But if we gave you a smaller unit, like part of the hemoglobin that makes up your blood, you have different chains within that hemoglobin. That would be a peptide. So peptides do not have their independent function, but a protein does. Egg white is a protein, for instance.
Which brings up a really important point to be made here. At some stage in all of our lives, we've been dealing with something very, very hot. You grab the pan, or I remember when I was a kid, man, let me tell you, I had a hot glue gun, and I was
I did that yesterday.
building something
Part
with
of my
hot glue,
favorites.
and you can burn yourself, all right? What happens in some of these cases, we can damage our tissues. What we're really doing is we're busting up these unique three-dimensional structures of these proteins. Proteins must maintain, like hemoglobin, must maintain, in that particular case, a big quaternary structure, multiple units, in these unique shapes that give them their function. Our enzymes that help us digest food is a very good example of this. And these unique shapes are held together with very weak hydrogen bonds, okay? A side product, if you will, of polar covalencies, all right? And these unique three-dimensional shapes, dealing with polypeptides, these shapes give proteins their functions. Now, we can test for the presence of peptide bonds, if you will. We can test for the presence of proteins. We can do this by using
Well, I say Biuret.
or potentially puree. You can go and Google search and figure out your own ways. But regardless.
Mine is a Latin version, and Mr. Hopper told me his is a French version, so you'll have to see. But regardless, it's this midnight blue solution. And notice that it's a different color than your Benedict's reagent. Okay, so we've got midnight blue here, which sometimes is hard to detect the change in color when it goes to violet. So sometimes, particularly on a lab test, you might want to hold this up, your test tube, up to a piece of white paper and figure out, are you really dealing with midnight blue, or are you dealing with violet? Violet is a purple violet. In that box of 100 crayons, it's not red violet, it's purple violet. So sometimes, particularly if you have some color deficiencies, color blindness, that you might have a little bit of trouble detecting the change from the midnight blue to the violet. But always white paper helps make it easier.
Now, a very important point to be made as we get towards the end of this little introduction is the importance of table 2.1 in your lab manual. Man, let me tell you, what this is going to
It
give
is
you.
on page
Mine's 24. This is the
24,
old
yeah.
version.
No, you're right, it's page 24. Just didn't have my book in the right place.
And what we have here is going to give you which macromolecule you're looking for, which test is utilized for that, and what the general outcome will be if those macromolecules are present. So this chart is very important.
And obviously that means on your lab test, you're going to
This
have to know this information.
Absolutely. You can guarantee, we'll throw a test tube up and it's going to have, a couple of layers, almost clear on the bottom and red on the top. We're going to say, number one, what test do you think this is dealing with? And that would be a
lipid test.
a nice Sudan, how about that? A lipid test. Part two, what reagent would be utilized to identify this?
Sudan 4.
And number three, I suppose, would be, is this a positive or a negative for that test? And in this case, it would be positive, and here it would be negative. And that, folks, is a nice introduction to macromolecules as labs. So let's jump into the actual experiments and see how they turn out. This is your first experiment for simple sugars. This is on page 25 of your lab manual. And what we have here in front of us are 4 solutions and 4 test tubes, all of which will be tested for simple sugars. And if you check your lab manual, we've got 4 test tubes, and those test tubes are supposed to be lined up as glucose, which is there. Onion juice, which is in the second place there. Starches, which is the third one there, as you can see here. And then, of course, distilled water. Now, remember, positive and negative controls. Positive, negative controls. A simple, where am I at? A simple sugar, generally speaking, is going to be something like
Glucose.
yes, indeed, something like
So
glucose.
that's a positive control because you know before you do this experiment that this solution contains glucose.
Absolutely, without any question. And then a nice negative control by comparison.
Well, what did we say before? You're really in trouble if there's anything in water besides water. So distilled water is our negative control today.
All right, so what we're going to do is we're going to follow our directions here. I forget exactly, let's see, 2 milliliters of each of the solution and 10 drops of Benedict's reagent into each tube. And then we're going to take those and place them in a hot water bath at roughly 80 degrees, which is what we have. It's hard to see here. Let's just see if I can get right in on it. Survey says I kind of can. And that is right around 90 degrees Celsius, which is just fine. That'll do. Karen, is that close enough?
Looks good. Remember, we're on page 25 and 26 in our lab book to carry out the Benedict's test.
All right, so we're going to set these test tubes up, and before we drop them in, we're going to come back. So what we've got here is our pipette. It's a good old-fashioned disposable. Where's 2 milliliters at?
Right here.
Right there. And you can see we've got roughly 2 milliliters in there. You can see the line on it. And that's going to be 2 milliliters going into our first, identified as number 1 test tube, right there.
Right there.
So there's 2 milliliters in. All right, let's go ahead and hit 2 milliliters in the next one just for identity's sake. So we're using a different pipette. Why are we using a different pipette?
Well, we don't want to contaminate the solutions because
Cross-contamination.
That's right. These tests are very sensitive. So if you had a little bit of glucose going into this tube, you might get a reaction because of the glucose.
Absolutely. So there is 2 milliliters of onion J. Now we're going to come back with all these full, and then we're going to look at what the reagent looks like going in. All right, what do you have in your hands there,
So
Miss Rose?
I've got our test tube of glucose, and we're going to add 10 drops of the Benedict's reagent.
Okay, which is blue in color, so let's see what it looks like.
Turquoise blue. So we're going to go 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. This is dropping fast today.
Let's take a look-see.
Okay.
It's got this nice, very pretty color to it. Yes, indeed. So we're going to put that one down, and then we're going to get the rest of them set up, and then we're going to look at it in the hot water bath. We've got our test tubes numbered 1 through 4. We're going to drop those in. You can see that one's nice and blue. This one here has already got a little bit of a weird shift going. We're going to place it in and see what happens. Here comes another one. Stir it up nicely. And there's number 4 going in.
So notice that the color, we're noting the color now, but the most important color is going to be the color after they are heated. This just kind of gives you an idea so you can see if there's a change and if it's enough to be a positive color.
So what we're doing now is we're waiting for 5 minutes. So we're going to note the time, and then we're going to come back to these. Wait a second, what's happening there now? Can you see that test tube shifting coloration? Now that's test tube number 1, which I believe is straight glucose. And Rose, could you remind us what glucose is?
Glucose is a simple sugar. We learned that in chapter 3.
And this is a test for simple sugars. So look at that amazing color change occurring. I wish you were here to see this in real time. It is really quite an effect. It's almost like a lava lamp. It's just boiling and roiling through. And you can see on our little experimental here, number 2, which I believe is onion juice, it's also got a little bit of a shift occurring. So this did not happen without the boiling of these fluids, but with the boiling of them, or placing them in a warm solution, you might say, we're seeing an effect. This is a change based off of heating these up with the appropriate solution, and look how red that's turning now. That's amazing. An amazing alteration. Whereas our good old-fashioned distilled water here
Hasn't changed yet. Remember, it's our negative control.
versus glucose, which was a positive control. Excellent. What we have here are our 4 test tubes. Now, I forget which ones are which.
Okay, so number 1 is glucose.
Which has turned a nice orangey red color, which if you look at your chart, okay, color change from pale blue to reddish orange, final color, or yellow or green, smaller quantities. The idea here is that nice orangey red color right there, that is guaranteed quantities of simple sugars, like glucose, that is literally glucose. And then we have number 2 here, this was onion juice. That has turned here in person, it looks pretty orange. Would you agree with that?
A Halloween orange.
Yeah, on the camera it looks pretty yellow.
So you can see not quite as intense as the red. So while both of these are positive, there are fewer simple sugars in the onion juice than in the glucose. And then notice from your table, green is positive also. So green just would have fewer simple sugars. So you can go green, yellow, orange, red, and then a really reddish brown. And that would be fewer sugars to more sugars.
And then our last 2?
Okay, number 3 is
Let's pull this out.
starch, sorry.
Starch. And then
And then we have distilled water.
now starch being a polysaccharide obviously did not have any kind of color change. All right.
And even though starch is made of simple sugars, they are not free simple sugars. And that's the idea. This test is for free simple sugars. In other words, unattached.
So they are
simple
bound,
sugars.
in other words. You could take that and you could treat it with an enzyme which breaks down starch, and it would then change colors, which is fascinating. And then, of course, we have number 4. This is our negative control, which is good old-fashioned distilled water. So it just looks like the reagent in water, which is literally what it is.
That's it.
All right, so there they are, 1, 2, 3, and 4. You can see 2 color changes. Not quite the same, but still positive. They don't all have to be identical. And then 2 without a color change.
Notice they don't look exactly the same, but they both are the negative color.
And that's got it. All right, folks. Our next test here is activity 2, a test for starch in the iodine reaction. What we have to have here is 5 test tubes. All right, what we have is glucose, maltose, starch. That's egg white, albumin. Okay, egg white, albumin. And distilled water over inside. That's 5 test tubes. And that's what we have here. 1, 2, 3, there's 4, and 5. Now, further, there's a second part to this experiment. And the second part is to take a piece of onion and a piece of potato, which is what we have here. Okay, we got some potato there, and we got onion on that side. And what we're going to do is we're going to add iodine, which is this kind of almost like a tea-looking liquid. All right, this kind of tea-esque liquid, just like that. And we're going to add it to these test tubes and see if there's an augmentation. Now, Miss Rose, what are we looking for?
Okay, so when there's a complex polysaccharide that would make the tube positive, the iodine is going to change from this golden brown color to a purple color. Now this is going to be a deep, intense purple.
All right, almost black.
Like a blue-black, yeah, or
Okay,
violet black.
so first things first. Here is test tube 1. That's glucose. Let's give that
get 10 drops.
Let's give that a shot and just see what happens.
Let me have
All right, here it comes. It's going in.
1, 2, 3. So there's your 10 drops.
And it's still a little like a tea color, okay? So I would call that a negative. Glucose, as it turns out, is not a polysaccharide, as we've established. It's a nice
And
monosaccharide.
so notice in this test, the color will change immediately. 3. So there's your 10 drops.
There's
We'll
maltose,
stir.
okay, maltose, which is a disaccharide, which is not starch, a polysaccharide. Okay, so this disaccharide stays a nice tea color, just like glucose.
So it's got to have multiple simple sugars, not just 2, in other words.
Right, and here we have starch. Now let's observe the process. Whoa, look at the change, okay? Very, very different. Remember, this is a
Here's your
test
10.
for polysaccharides. What we're looking for is a big honking starch molecule, and this is literally straight up starch.
So see against the white paper, see how dark that is?
And you can see that kind of a blue on the side there. Okay, so this is a nice blue-black, from my
Absolutely.
perspective, a very nice starch reaction. Yes, indeed. All right, so our next one is albumin, which is in essence egg white. and it stays a nice tea color. One of the good things
Here's your
about an
10.
iodine reaction
There's your 10 drops.
is it's very obvious. Okay, nice 10 drops, gives you that tea color, not unlike these over here. And then last but not least, we have distilled water, which is our negative control. And nothing is
There you
happening.
go.
All right, so that is a nice set of starch reactions. Let's check out our potato and our onion. Now, here they are. Now, remind us which one's which.
Okay, so the idea is we're just going to drop some iodine on top to see if there's a color change. And of course, on the left is the potato, and on the right is the onion.
And again, the glory of iodine is it's a very obvious reaction. Here we go, potato, wow. I mean,
All right, that's just one drop.
that's one drop. It wants probably more than that, but I think that's pretty conclusive. You're going to give us another one just for fun?
Yeah, drop or two. Okay, here we go.
Wow, I mean, complete darkness versus the onion. Wow, virtually nothing.
So this shows you that you don't have to always have test tubes and things. You can use the vegetable itself and do your test.
Or any solid for that matter
That's
that
right,
one has
that's
to
right.
experiment upon to identify the presence of
That's right.
certain macromolecules. And it also tells us a little something about a potato versus an onion, and that is
That's
the
right.
potatoes quite obviously make starch as its polysaccharide, whereas an onion, though a plant, though forming a large underground bulb, as it were, is not making large quantities of starch.
So you have a question in your lab book that asks you how, look over here, says, it's number 3 and 4, do potatoes and onions, are they positive for starch, and how do they store their energy? What they're getting at, it's not just a repeat of the first question, they're making the point that Mr. Hopper just mentioned, is that vegetables and trees and fruits and all kinds of plant material have to store their carbon and energy somehow. So the question is, how do they do it? And you see from this test, potatoes store their carbon and energy as starch. Now, the second question is, and this, I know it's not in your lab book, but you see that onions do not store carbon and energy as starch, okay? Onions do not store starch. So how does this plant material actually store their carbon and energy? Okay, now, you did not test onions in this test, so you can't say they don't automatically store starch because potatoes do, but you have to go back to your Benedict's test. And remember on your Benedict's test, you did onion juice, okay, and you got a positive color for simple sugars. So if they don't store starch, you know they store simple sugars. Those of you who cook and eat grilled onions, you probably know what I'm talking about. Grilled onions have a sweet taste to them, and that's because they store simple sugars.
Our next experiment is going to be for proteins. This is the Burey's reaction. Now recall that our fluid here, our fluid is a nice, what would you call that?
I call
Miss
it
Rose?
midnight blue.
Nice midnight blue to it. And what we're trying to see is if this has the presence of proteins, of peptide bonds, if you will, we're going to find a deeper purple.
Right,
All
more
right.
like a purple violet.
So we're going to do these drops. Let's put them over white so we can really see it go.
Okay.
All right, so what we have, number 1, this is,
Here's our starch.
all right, starch. Now recall, starch is a polysaccharide. Ergo, we're not expecting to see anything. This could be considered a negative control in the grand scheme.
Okay, and then number 2 is albumin, which is
Albumin.
egg white.
Indeed. Protein rich. And then, of course, good old-fashioned distilled water, guaranteed nothing in it. That would be a
That's really your
hardcore negative control.
So even though you know a little bit more about starch, technically that's your test sample today.
All right, let's see what we got.
Okay.
Number 1, so here we have
Here's
starch.
our 10 drops.
Can you hold it down here
Okay,
over the
sure.
white? Yeah, now we can really see it. So you can see that there's kind of this bluish color going in.
All right, so we need to mix a little bit.
All right,
Swirl it.
here we go, perfect. Nice lighter blue, which is just from less fluid being in there with a clearish fluid.
Sure.
All right.
Same color.
So I'm calling that negative.
Okay, here's our albumin.
All right, albumin. Here comes the reactions. It's going in, a little bit of shake to it. Now what I want you to notice is that we put the same stuff in these. But when you look at this, like as I'm sitting here in person, that is purple.
That's right.
Okay, it looks kind of bluish, but probably because this is behind it. But I'm here to tell you, folks, that right there, that is purple. As purple as the day is long.
Like we
Nice.
said, if you have any
Really?
trouble with seeing the color, you can
See, look at that on the
lay it
side
out.
here.
That's a good thing to do, and put it against white paper.
I mean, that is purple. Again, as I sit here in person, it may not show up on the camera, but that is purple. Purple in person.
So what we want you to do to be able to see the difference, since you're not in lab where you've got, you're doing this yourself, but that's on the
Can you hold them
left.
both, if you don't mind?
Yeah, sure.
Heavy camera.
So on the left is the albumin for the Biuret test, but on the right is the starch test. So you can see these are different colors. You've got more of a blue-black, even though sometimes depending on the food substance, this could be a brown-black or a purple-black, but you see it's got the more blackish color to it than what the Biuret test
I
does.
feel that transparency really falls out too.
Yeah.
Like you can see the starch is very dark. You can't see
Yeah,
through that at all.
that's right.
Whereas here, this purple is very transparent indeed.
Yeah, so those are just those two tests side by side, so that if you saw tests, on a lab test, for instance, where you see tubes from different tests, you know the difference in what a starch test will look like, a positive starch and a positive protein test.
Awesome. Okay, and then we got one more. Let's see a negative control here.
Okay, so here's our water. We're going to add the 10 drops. All right, here goes 10. And we're going to mix.
Nice. Compared to the first tube that we did here.
That's right, compared to the water. And then again, here's your positive protein test.
So 2, which are negative, that being starch and water, and then 1 positive being albumin. Excellent.
Okay.
And that, folks, that is the Biuret reaction. Or Biuret.
Either way.
All right, folks, here we are on activity 4, testing lipids using Sedan 4 test. Now, what we have done is we have gone through with our 4 test tubes. We have one that is for glucose, one that is for potato juice, one that is for oil, and then one that is for water. And what it asks us to do, as you can see I have a star here, is we have gone through and we have added 2 milliliters of water to each tube.
You might wonder, why do we have to add water to the tubes? But remember, oil, for instance, if you have a positive lipid test, then, and it has to separate away from the polar water, how's it going to separate if you don't have any water in there? Now, you can fudge and say you've got an oil test tube, just put it in there. But you have to, this is science, you have to treat all test tubes the same. So even the water test tube that had 2 mils of water in it has an additional 2 mils of water because every other test tube was treated that way.
And mind you, what we're trying to do here is get at the concept that in a laboratory setting, say you're working for an environmental lab, and they were asking you to identify the presence of certain chemicals within a soil sediment sample or what have you, anything along those lines, everything has to be treated identically for continuity. All right, we have to control where everything is and what's happening where. So now then, let's add some.
Sedan 4, and we add
So this
10
is a nice
drops.
red color to it already. So let's see what happens here.
Okay, now with these, you have to really shake, okay? So I've put my thumb over the top, and we're gonna really mix, okay?
And what we end up with here is a nice sort of reddish fluid.
Right. So notice that even though it's red, and you're seeing in your lab book red is positive, notice there's no separation of layers here. It's a consistent one color throughout.
Because what we're looking at is glucose, which is a simple sugar, a monosaccharide, not a lipid.
All right,
Next.
so here's our potato juice. 1, 2, 3, 4.
Every fiber of my being, I'm fighting off the urge to say potato J.
Okay. All right, so let's give this a good shake.
And
And you see, not quite as red, but still is consistent throughout. So it's one layer and has the pinkish reddish color.
indeed.
Okay. All right,
Next.
so now we're to our oil. All right, so 1, 2. Okay.
All right.
All right, so good shake here, just like you'd shake up salad dressing.
And we end up with
All right,
this kind of a milky coloration, but the trick is what, Miss Rose?
So you can see still the pinkish reddish color, but can you start to see the separation? Okay, there are little bubbles here that we can tell that the oil, just like in salad dressing, how you have oil and vinegar.
It's looking real clear there on the bottom.
Yeah. So we're going to come back to this one in just a minute.
The trick is that you got to let it sit.
That's right.
Yeah. Let it sit for a little bit.
In lab, if it sits half an hour or an hour, that's really the better thing.
You can really see on the bottom of that.
Yeah, you can see.
That there's, just hold it still there. Let's see if I can get a good shot. And you can really see it getting clear on the bottom, and then it's got a layer at the top. That's giving us a little bit of an indication at this stage. All right, let's do the next one, then we'll come back.
Yeah. Okay, so here's water.
So negative control.
Okay. All right, so let's give this one a good shake. And you can see pink throughout. So even though the color is very similar, okay, there's no layering to the water.
All right, let's
Okay.
put them side by side.
And let's come back to the oil. See how you can see even more of a separation now that it's set just a little bit. And that will continue to progress over time.
And we'll put a picture of that in at the end as well.
Okay.
All right,
Sounds
perfect.
good.
Perfect. And that's how you run a test for lipids. What we're trying to see, again, just to make it very clear for us, is you can really tell now we're getting a separation out of layers. You can see that the bottom of that is almost clear, and the rest of it is not. Okay, it is not. So that is a good indication of a positive Sudan test.
Okay, so the last activity in Lab 2 is to check out 2 unknown solutions. All right, we just have some food substance here. We have no idea what this is. And to see what chemicals are in it, and if we can predict by virtue of the positive test versus the negative test, what that food item might be. So even though y'all don't know what it is, we do have some kind of idea since we had to make up the solutions. So we have taken 4 test tubes for unknown number 1.
1, 1, 1, 1.
And this represents the 4 different tests that we do, the Benedict's, the starch test, or complex polysaccharide test, the protein test, and the lipid test. And we're going to do the same thing with unknown number 2.
2, 2, 2, 2.
And so we're going to see if we can figure out. And Mr. Hopper is going to
Give you an idea.
We're going to do a little smelling, and we're not going to do any tasting, but we're going to see if we can. So he's going to make some observations to you.
Doing our unknowns, what you need to do is you need to be on page 33 here and fill in this chart as you observe these experiments. Okay, samples 1, 2, 3, and 4. We're just doing 2 at this stage. So this could be sample 1, this is sample 2. And here is what you observe from a Benedict's reaction. Here's what you observe from the iodine test. Here's what you observe from a Biuret or Buray's test. And here's what you observe from the Sudan 4.
So fill in unknown number 1 all the way across the page, and then unknown number 2 all the way across the page.
Perfect. Okay. And that's how you're going to figure out what the unknowns might be.
What they might be.
Number 1, it's got a, like whatever this is, it's pretty consistent. Okay, it's got a consistent appearance to it. It's, it's, I can see little bits of particulate in there, but it's not like there's a bunch of junk floating around. It smells real sweet. Like this has a very sweet smell to it. Like if you were to put this on top of some vanilla ice cream, it would taste delicious. Okay, whatever it is, Lord knows. All right, but I'm just telling you, it's kind of thickish, and it definitely smells good. Like it's appeasing. It's like, again, there's dessert to this. I don't know what, but dessert to it, and a little bit of particulate. You can see how it kind of gets on the glass there. The second one. This is quite particulate. Okay, this is full of stuff is the way I would describe this scientifically. Stuff. There's stuff in here floating around. It definitely gets on the glass. You can see how it gets on the glass here. And this is, I mean, it doesn't smell, it smells like food. There's definitely a food scent to this, but it's not that appealing. Like, it's very drab. Bland. It's very bland, but definitely particulate. Like you can tell there's a little bit of dissolving into the water, but when I hold this, it really settles out. It wants to settle. So it's very particulate. It's very grainy, for lack of better terminology.
Well, since both of these are food substances, we know they're safe, they're something we would consume. We can smell them like you would normally smell something. For those of you who've had chemistry lab experience, you know we're really supposed to waft things and use our hand to bring the scent up to our nose. So we cheated a little bit today because of the type of item we use. But if we were using chemicals like maltose and things like that, we would have to follow the chemistry safety technique.
The idea is, if it's something potentially dangerous, you don't want to directly smell it.
That's right.
We're being safe because, again, if we're in like an intense environmental lab or something, we're going to be far more careful than this. This is just good old-fashioned biolab. So I believe it's time to add some reagents.
Benedict's reagent first.
All right.
And I'm going to do test tube 1 and 2. So remember, there are 2 different unknowns. And it gets 10 drops.
All right, I'm going to come in there and get seated.
Okay, and of course, we're going to have to heat this one.
All right, let's see it up front here.
Let's have a good mix. So
And again, what do we have to do here?
we're going to heat this in a hot water bath for 5 minutes. But these are the initial colors.
All right. All right, here we are at the hot water bath. And let's just see if we get any augmentation. All right, here we are after 5 minutes. And what we have is the first test tube number 1, there. All right. It has definitely taken on an orange coloration, and test tube 2 has as well. Now, if you're not here, this is much more of a reddish orange, and this is more of a burnt orange, kind of a light orange coloration to it. This one was much slower. This one was much more intensive from my perspective. Ms. Rose, would you agree?
I would agree.
All right. And that's what we've got for, which experiment are we doing?
We're doing our food composition, our unknowns. And before we go any further.
1 and 2.
1 and 2. Before we go any further, we should have said this in the beginning, excuse me. But you notice that they have you develop a hypothesis. So the idea is we gave you some consistency and smell observations, and based on that, what would you think these food substances, what would they be? You don't get to make a hypothesis after you do the experiments. So before the experiments, what did you think those food items, what would they be? So.
All right. And again, here we're looking at simple sugars. Simple sugars.
Yes, Benedict's test.
They are both positive, but from my perspective, the second one is a little more intensive than is the first.
Absolutely.
All right.
All right, so we're set for the iodine test for starch and complex polysaccharides, and we're going to add 10 drops. So we've got 1, 2.
Again, our goal is to look for a dark color change, and that's pretty apparent right there, folks. Certainly a presence of starch there. Wow, look at that.
That's unknown number 1.
Wow, very dark coloration.
Okay, let's do unknown number 2. All right, here goes our 10 drops. Okay, there's 10.
Telling,
Okay.
very telling.
Oh,
Oh, a little bit.
so see how mixing becomes important.
Wow, look at that.
Wow, I didn't
So
think that was
I
gonna
didn't either,
change.
really. So, frankly, I would say that probably doesn't contain the amount of starch as does the first.
Yeah.
So that first one has lots of starch in it. And as I'm sitting here in person, folks, it's hard to see that on the camera, but the first one here, number 1, is hang on, this one's okay. The first one here, number 1, is very dark indeed, and the second one here, number two, is not as dark from my perspective. Would you agree with that, Ms.
Right,
Rose?
it's not as dark through the solution.
All right, so number 1 has lots of starch from my perspective, and number 2 would have less,
Yes.
which, considering what we saw in our previous experiments, is telling. It tells us something. All right, and that is our test for starch using iodine. What are we doing?
All right, we're doing the Biuret test for protein. So we've got unknown number 1. We're going to add our 10 drops. So we're going to go 1, 2, 3. All right, there's 10 drops. Oops.
Mixing is important.
Okay.
What do you see?
Now, I see a gray color. And some of you are going to say this is positive because it has a color change. But the question is it a color change to the positive color? And my thoughts are that this is actually not purple enough to be positive. This is more of a gray color. So I might reserve comment till after I see number 2. What we should have done was keep our controls, and then we could put the controls to either side and then see if this sample is closer to the negative control or the positive control.
All right, let's hit the next one, see what happens.
Okay. All right, so we'll let the jury be out on that one. All right. Okay. So I would say a definite negative here because it's blue and
It's
not
quite
purple.
blue.
Yeah.
Quite blue.
Yeah, even more blue than what the Biuret reagent itself is. And since we don't have controls, let's just put number 1 up by the Biuret reagent. And I don't really, do you see any purple?
I don't see any purple.
Yeah, I don't see any purple either.
I would almost call both of these negatives.
Yeah, so you can see there definitely was a slight color change. So color changes don't automatically mean positive. You got to look at the actual color. Is it closer to the negative or closer to the positive?
So I'm going to call both of these negative
Absolutely.
for proteins,
Yep.
which is a result in and of itself. Being negative is not a problem.
No.
Being negative is a result.
Okay.
All right, let's run the next one. All right, last but not least, here we have a Sudan test for lipids. We've already added our water to these, and here comes the Sudan itself. Look at that.
Okay. All right, let's shake this one up. Okay. All right, so the question is, do we get layer separation, not just because of the particles that are within the sediment that's within the solutions?
So I tell you what we could do is run the next one.
Yeah,
Let that one
let's
sit.
see. Let's let that one sit. Okay, let's shake this one up good.
All right, so what we can do is we're going to let these sit for a little while, and then we'll come back to you after a few minutes, and we'll see if we have any good layering. But if you look at them as they sit right now, we have a nice, even, kind of pinkish color. Would you agree with that? Pinkish. And we'll come back in a few minutes once they've had time to sit and rest and see if we come back with any layering. Okay, folks, what we have here is the conclusion of our Sudan test. Now, this is our old Sudan that we did as a positive control a while back, and we held on to that for this very occasion. You can see it's a clear double layer here, whereas over here, these do not have a clear double layer. Now, this one, let me grab it from you. This one does have something happening in there. There's something going on. It's hard to say if it's just particulate. This is number 1, by the way. It's hard to say if that's just particulate from the food stuff that we were using, or if that's some sort of effect from the Sudan. But I'm inclined to say that if that's anything, it's very little. Very little. Ms. Rose, what's your opinion on the subject?
I agree with you. And number 2 is obviously negative because you can see how it's very clear. Even though some particulate has settled out, there's no layering.
Number 2 is
And it's a very clear pink.
obviously not
Yeah.
positive for lipids.
Right.
Number 1 is hairy. Like if there's lipid in there, it's not much.
That's right.
All right. Whereas our old positive control, you can clearly see there's 2 layers here. It's almost transparent on the bottom and quite red at the top.
Yes. And that's the oil.
That's a
in the-
very obvious oil
from the
reaction.
Sudan
All right.
test.
So that's our Sudan test. All right.
Okay. So now that we have concluded the testing of the 2 unknown food samples, and you should be writing results in your lab book here, as we explained in the beginning, we have given you the consistency and the smell. And so based on what's present and what's absent as far as our organic compounds, what do you think is in our unknown solutions? What do you think they're made of? So we're not going to tell you today. As you go to fill in your lab report that you're going to be turning in, we want you to give us that answer, and just for fun, we'll see if you're right or not, and we'll be happy to share it at that point in time.
The idea is come up with something
That's right.
and, just make sure you're in the hunt.
That's right.
That's the goal. Be educated guessing. Don't say you think it's ground up steak when it's full of sugars and no protein.
That's right. All right. So no harm done on that, but we want you to put your scientific brain to work and see if your hypothesis was proven true or false.
All right. And that is lab
Exercise 2.
That's it. Macromolecules. Hope you enjoyed it. All right, say adios.
All right, goodbye, adios.